That was incredible.
I'm sorry it was so long.
Thank you. Yes, our next speaker today is Dr. Zachary Manzer from Cornell University. He'll be talking about creating biomimetic interfaces using the cell-free synthesis of transmembrane proteins. Yes, it's a 15-minute talk plus 5 minutes of questions. So when you're ready, Dr. Manzer, please start.
Sure. Can you see my screen okay?
Yes, we can.
Okay, great. Yeah, thank you for the introduction. And thanks to the organizers for inviting me to this. It's really been an enjoyable seminar series each week. There's been a lot of interesting talks, so I'm happy to contribute mine. So as Peter said, today I'll be talking about sort of a new project in our lab that we've just been working on recently with in collaboration with the Kamat Lab at Northwestern University. And basically what we're doing is using cell-free synthesis of transmembrane proteins in order to create new biomimetic interfaces that we can use for synthetic cell applications in biotechnology. So before I get started, I just want to orient you sort of where we are. We're at Cornell University. It's this school in upstate New York, in Ithaca, New York. And it's a really beautiful place. I'd really encourage you to come visit if you ever get a chance once things are a little more normalized. But yeah, so when thinking about the important components in cells, one of the most important features is the interface of the membrane. In bacterial cells is the outer membrane. In eukaryotic cells, obviously, there's the plasma membrane of those organelles. And these membranes are a really critical interface in maintaining interactions and maintaining cellular viability. They have immense complexity. You can see the little schematic I have here. They have glycans that are added on either to the lipids or the membrane proteins, which modulate functions and transmit signals. The lipids themselves have the hydrophobic interiors in the tails and the hydrophilic exteriors in the head groups. And a class of molecules that we're really interested in is membrane proteins, which can be lipid length, they can be transmembrane, they can be peripheral or membrane associated proteins. And these all work together to carry out really complex functions. And I've listed a few of them here. They can carry out enzymatic conversions in the cell. They can serve as receptors to transmit information across the cell membrane, as well as serving as a transport species across the membrane, such as with ions or other electrolytes. And so because these are so important, scientists and engineers have really been working how we can create synthetic versions of these. And you can think that if we can capture these things that transmit species through, they can be used for separations or maybe for biosensor applications. Enzymes that carry out reactions can be used to create defined planar chemistry. An example I've shown here is with glycosylation, which is an immensely diverse and complex reaction that takes place inside cells. And finally, anything that transmits information can be used to sort of screen therapeutics. And this might be a way to sort of screen membrane proteins involved in pathogen detection or looking for drug discovery applications. These are just a few examples of ways we can sort of use and study membrane proteins in biotechnology or building up synthetic cells. And they all really rely on the proper incorporation of membrane proteins into a lipid environment. And they'll require tunability to maintain proper performance in any system you want to design. One of the bottlenecks in really achieving this is that our current analytical tools are sort of limited in their capacity. One of the first major ways people study these is in cell-based assays, in which you basically take a live cell, image things that are going on inside it, and this can be very powerful. But unfortunately, you're limited to sort of the complexity that you're getting just inherently in the membrane, as well as the constraints of being able to engineer and genetically modify these organisms to see the response that you want to see. The other side of the coin is a lot of people are familiar with here is in model membranes. These can be a variety of different approaches. Detergent-mediated micelles, nanodiscs, which are lipid membranes belted by proteins or small molecules or polymers, as well as liposomes, which are spherical bilayer structures. These all pose certain advantages in studying membrane proteins, but unfortunately, a lot of them have difficulties in purifying proteins if you're doing a reconstitution approach. If you're doing a cell-free approach, a lot of them have issues with stability. And also nanodiscs potentially don't have a defined orientation. So if you're trying to do a flux-based assay, you aren't able to sort of get differentiation between both sides of the membrane. In liposomes, you're unable to access the interior. So you're sort of limited based on what you can do with the exterior of the membrane. That's not to say that these aren't really powerful tools in synthetic cells, but this really motivates us into sort of how we can sort of extend this into something that's more robust and more stable to further the applications. And one way that we do this in our lab is using a supported bilayer system. What this is sort of just like it sounds like. It's a supported membrane on a planar surface. And this provides a lot greater stability. And because of the geometry, it's much more compatible with surface techniques to do spectroscopic studies, optical studies, electrical studies, et cetera. These have really gone through revolutions since they were first introduced in the 80s, up to the point where we can now take fully native membrane materials and incorporate them into these bilayers. One of the issues with these is that you still have the complexity. And so if you want to get isolated effects, what you currently need to do is reconstitute proteins in these bilayers, which unfortunately has the downsides that I mentioned previously. And so what we want to do ultimately is be able to put a defined interface in these planar supports that gives us all of the ability to sort of use these planar bilayers with their greater stability and access to analytical tools, but have a defined interface without the complexity needed, so we can build up these synthetic platforms from the bottom up. And so this led us to the question of whether we can directly incorporate membrane proteins into support lipid bilayers. And this led to a publication that we recently got out in ACS Applied Biomaterials. I'd highly encourage you all to check it out if you haven't already. And what we do here is we form a hybrid support lipid bilayer made of lipids and diblock copolymers and use cell-free synthesis, which I know a lot of us are familiar with, but very briefly, you can isolate the machinery for protein synthesis outside of a cell, either in a purified format or in a cell lysate format. Add in a piece of DNA of interest, and then you can directly incorporate it on demand into your membrane environment. In this case, we're using the model membrane protein, the large mechanosensitive channel of conductance from E. coli. This is a 10-pass transmembrane channel, and we include GFP as a reporter of membrane folding, protein folding, as well as the ability to image these proteins after expression. And we envision that the ability to create these mimetic membranes with functional and oriented membrane proteins will provide us a way to have a platform to carry out sensitive and precise protein events. So as I mentioned, we're using a blended membrane system, and this comes from the basis from the Kamat lab at Northwestern University. They published in 2019, in which they can form blended membranes using a diblock copolymer. In this case, it's polyethylene oxide polybutadiene with the OPC lipids. And they found that by blending these diblock copolymers and lipids, they can tune the biophysical properties of the vesicles and affect the protein synthesis and properties. So the first thing we did was take a little look at the vesicles themselves and make sure that the diblock copolymers and the lipids were interacting like we'd expect them to. And using dynamic light scattering, what we see is that as we increase our mole percent copolymer, we're largely keeping the same diameter and we're largely keeping the same zeta potential. And this indicates to us that any changes in the membrane properties in the supported bilayers will be from the actual ratio of the membrane components and not just from the diblock copolymer themselves. And so now that we have these vesicles, what we want to do is form them as supported lipid bilayers. And the first thing we want to study is the 2D lipid fluidity. The ability for lipids to move around is an important biological property that cells can maintain, and we want to see if we can maintain it in our system here. And the way we study this is that we can incorporate fluorescent lipids into this membrane, and we bleach a small spot with a laser. If the lipids can move, the bleached ones will be able to diffuse away, and the unbleached ones will diffuse back in. And what you see is a recovery over time if the lipids are mobile. We see the same behavior in 100% GFPC and in the 15% copolymer. As we go to 50% copolymer, what you see is that the bleach spot does not recover, indicating these lipids cannot move. We can track these over time. In the left graph, we just have the normalized intensity as a function of time. You can see that the behavior is largely the same as we increase in percent copolymer. But when you actually calculate the diffusivity, what we're seeing is that we're getting a steady decrease in diffusivity up until 50%, which we no longer see mobility. This might mean that as you increase percent copolymer, the polymer is actually preventing lipids from moving. But because we do maintain a very high mobile fraction of lipids, what we believe is happening is that the membrane viscosity is actually increasing to a point where the lipids are just becoming too viscous to move through each other. The other alternative is that the vesicles might not be rupturing, and so we wanted to take a secondary approach and study this using coarse crystal microgrounds with dissipation. In the interest of time, I won't go in depth with the actual details here, but basically what it is that you have a silica-coated sensor you can deposit materials on, in this case vesicles, and you can read out changes in the frequency and energy dissipation from the surface, which gives you indications of essentially mass and the rigidity of your surface. So what we care about is the final change in frequency and dissipation. And what we find is that as we increase percent copolymer, we're seeing a pretty standard increase in the frequency, which corresponds to the increased mass of the polymer, and a very large jump at 100%, which indicates to us that these are unruptured vesicles because they contain buffer associated with the membrane. We see this in the energy dissipation as well. We see standard increases in the energy dissipation up to 50% copolymer, This is because we believe the polymer is giving us more flexibility in the film and a very large increase at 100%, which indicates unruptured vesicles to us. So we believe that up to 50%, we are rupturing vesicles into supported membrane, but at 50%, they're no longer mobile on the surface. So being able to tune this membrane viscosity is a nice feature of this system, because many times you want lipids to be mobile, but sometimes you might want to restrict diffusion without having to add additional biological components. So it gives us another synthetic tool to sort of control what's happening to the membrane. So obviously now that we can form bilayers, the next thing we want to be able to do is put proteins into them to carry out our functional assays. And we do this using two different approaches to give us more flexibility. The first is that we express the protein into a vesicle. We then rupture them on the surface. And the second, we form the bilayer first and then express the protein directly into it. And both of these should give us the same result. The first thing we check using these methods is whether or not the protein can affect the lipid mobility. And what we find largely is that there's not a huge difference between with and without protein. We see this both in the diffusivity and in the mobile fraction of lipids. So it indicates that the protein is able to incorporate and not have a deleterious effect on the membrane itself. An important thing we want to check is whether or not these proteins are oriented. They can be synthesized either inside out or right side up. And we want to see if that once they rupture, whether the protein is facing up or facing down. This is important when you want to do functional readouts, is that your protein should be oriented, so you know what side of the membrane is facing up, and you know if you're getting the right flex of your components. If the protein is facing up, we have a TEV cleavage site attached to it, and we can use the protease to cut this off. If the protein is facing down, the protease should not be able to access it, indicating that the signal should remain. And so what you see in the bottom left there, we express our protein and we see an increase in the fluorescent signal. After we add the protease, we see a decrease. This indicates to us that the protein is actually oriented upwards. And by quantifying this, you can see that we get about 90% reduction in protein. So they're all oriented very uniformly. We then repeated this with our second approach. We formed our bilayers first and then expressed the proteins directly into the membrane. And again, you see that before and after the protease addition, we see a large reduction in the GFP signal, indicating that we do have oriented proteins in the system. In this case, we're actually seeing an even larger reduction, which we believe is because we're not actually rupturing protein vesicles. And so there's nothing that can get stuck into the membrane. So the surface is actually much cleaner. Now, the orientation is very important for functional readouts. An important biological property that I haven't mentioned yet is actually protein mobility. The proteins need to be able to orient themselves in the membrane and associate with their neighbors. And so we want to see if we can maintain that mobility in our platform. And what we found is that you actually need at least 25% copolymer for mobility. And the copolymer is essential for this. And we believe it's providing a cushioning effect from the substrate. And so what we did is record these images. And I'll highlight one protein here, just to make it easier to see. And what you can see is that as this protein is able to diffuse around, interact with its neighbors. So we can take all these videos and analyze the motion of each of these particles using single molecule tracking. And what we find is that if you can look at some example trajectories here, we get diffusivities around 0.16 microns squared per second, and we maintain about 50% mobility of our protein. We see similar results in approach number 2, and that our diffusivity is largely the same across each sample, and the mobile fraction remains about the same as well. So we then wanted to extend this approach. Now that we know the lipids are mobile, the proteins are mobile, and they're oriented, we wanted to showcase some of the biophysical characteristics you can get using this approach. And what we did is a subunit photobleaching experiment to identify the oligomerization of the MSCL. Recall that this is a 10-pass channel, and so this will actually be a pentamer, and there should be 5 GFP per molecule if it's properly associated. And what you can do is, in this example here, track the fluorescence of a single particle. And what you see is it bleaches over time. You see single steps of where each GFP bleaches out. You can then calculate this over all the proteins you're imaging. And what we see is a distribution that corresponds to what we'd expect to see for a pentamer, indicating that these proteins are fully assembled. So now the work that we're extending this into and that we're really excited about is whether or not we can sort of now couple these membranes with active readouts. And so what we're doing is coupling this as a bioelectronic platform. And here what we're doing is using P.PSS film with supported bilayer on top of it. And this lets us do electrical impedance spectroscopy measurements and other electrical readouts in order to characterize these transmembrane proteins, whether or not they're functional or not. And so we perform the same protocol. We use direct self reinsertion of the MSCL channel into the membrane. We want to see if we can actually read out drug activation to see if they're functional. Here I just show the structure of the MSCL. to orient you what's going on. This is a mechanosensitive channel, and typically it's closed. You can have a chemically activated mutant, G22C, in which case you get this molecule empty set, which should open the channel and allow membrane or ions to pass through. And so, in interest of time, I'll just show the data here. What you're looking at is a Nyquist plot showing the changes in impedance. The important characteristic to get out of this is that the radius of this hemicircle corresponds to membrane resistance. And so as you get a larger hemicircle and a larger radius, you're seeing a larger resistance of ion flow through the membrane. And so we start with a base polymer here. We then add the bilayer, which then blocks the flow of ions into the surface. You see an increase in resistance. We then add protein expression. And for the wild type MSCL, when we add the drug, we see a very slight increase. We believe this is because the empty set is positively charged. And so there can be some slight interactions there that can block ion flow. But importantly, when you express the G22C, what we see is a decrease in resistance, which indicates to us that the empty set is actually opening up the channel and allowing ions to pass through, indicating that these proteins are inserted functionally into the membrane. So in conclusion, we are able to take diblock copolymer hybrid liposomes and form supported bilayers with tunable biophysical properties from them. We can then use cell-free expression to quickly incorporate membrane proteins into these bilayers. And importantly, the copolymer is essential for mobility. And these expressed proteins are oriented, mobile, and functional, as we were able to see with our activity readouts. And we really think this approach can lead to the ability to address a lot of different challenges in synthetic biology and building up synthetic cells, such as in transport phenomena, membrane protein biosensors, enzymatic conversions, and reading out complex binding events. So that I'd like to thank my advisor, Susan Daniel, my co-author on this project, Sergey Ghosh, as well as our collaborators, Northwestern, Neha Kamat and Miranda Jacobs. And with that, I'd like to take any questions.
Thank you, Dr. Manzer. Yeah, great talk. I'll have a couple of seconds for questions to arrive. Yeah, I might open the questions then with, so I was just curious to see, so if this is I'm not particularly familiar with the sort of with the supported bilayer systems in general. So I was wondering, what are the constraints on the size of the protein that you can get on the support surface? And I'm particularly thinking about proteins with large domains that might protrude in both directions. And obviously that obviously plays into the directionality of the insertion. But I was wondering if you could just maybe expand on the constraints there.
Yeah, sure. No, great question. I'll skip back to this slide here. It kind of shows what happens. So in a normal supported bilayer, you have about a 1 nanometer water gap there, which is obviously very small. And so what we see happening is that we think the copolymer is actually giving sort of a cushion to lift it off the membrane, and that's why we get mobility with the copolymer and not without. The size will basically be, I think it will be kind of protein dependent. Obviously, if you have a large cytoplasmic region, you can have a lot of interactions with the glass support. And so I think it'll really depend on each protein how much interacts with the support. You can have a larger cushioning system to sort of give you even more lift off the surface, because it's largely a physical interaction that's putting the motion and not sort of just a biological interaction. There are other supported systems using more tethered approaches that give you larger distances, so you can incorporate larger proteins as well.
Okay, thank you. So we have a question from Aishwarya Juneja. Is there a scope of lateral diffusion or flipping of molecules in the designed membrane? I think that might be to do with transmembrane, membrane translocation. Is that correct?
Sure. Yeah. So we haven't actually thought about whether or not the proteins can flip over. We've seen that they're all oriented in this sort of same direction, and that likely comes from the way the proteins are synthesized. Whether or not you can flip a protein over, my guess is that you would not be able to flip protein over the system. There are ways that you can inverse the vesicles themselves if you want to build it sort of in an inverse platform. But if I'm understanding the question right, I don't think we'd be able to sort of get a protein to flip over when it's already formed in the bilayer.
Okay, good. So Felipe Quiroz asks, are the ribosomes somehow directed to interact with the membranes to favor insertion of the transmembrane domains?
Yeah, that's actually a great question. It's one I've thought about a lot. In this system, we're actually using the pure system, so there's not chaperones and chaperones and translocons available in the system. So in our system, it is just the ribosomes being directed to the membrane. Whether the ribosomes, I think it's a couple of interactions between the nascent peptide coming out, wanting to go into a hydrophobic region. So I believe that is the thermodynamics are driving interaction there. But there are other people have looked at ways you can use start incorporating chaperones and translocons in order to get directed insertion of these proteins into membranes. But yeah, it's actually a really interesting question.
Okay, Alan Kay asks, extension of the question I was asking was, how large can you make the gap with a copolymer? What are the limits there?
Yeah, so that will come down to basically the size. Just to show like some basic theory here. This comes from polymer cushioning from Dijon. And this basically corresponds to the brush regime of what the polymer length is. You can imagine that as you increase the peg chain, you're getting a larger and larger cushion. You're obviously trading off some benefit, or there's a trade-off between the protein mobility and lipid mobility as you get to these large peg molecules. We've done in other systems up to peg 5,000, so you can get upwards of 5 to 10 nanometer cushions there. In this case, the peg group is only actually 600 Dalton, and so it's only roughly 1 to 2 extra nanometers. And so you have to play around with how much you can incorporate in there. Like I mentioned, there are some other more advanced techniques that you can sort of get more tethered systems and not just a generic polymer cushion. But roughly, you're looking probably, I'd say, around 10 to 20 nanometers maximum limit with this sort of system.
Okay, so we have time for maybe 1 or 2 more questions. So Christy asks, have you got a chance to do the similar cell-free protein synthesis into cell-sized giant liposomes?
Sure. We haven't done anything that large. We're using 100 nanometer vesicles just because that's where the range you need to use to form supported bilayers. Once you get above, I think, about 400 nanometers, they won't spontaneously rupture on their own. In this system, once the vesicles interact with the hydrophilic glass substrate, they'll just kind of burst into the supported bilayer. Once you get to a certain size, they won't rupture. So no, we haven't looked into cells that or vesicles that large.
I think we'll have time for one last question. So Hussein Mogimian Naval, thanks for your great talk. What do you think is the surface density of incorporated proteins in your bilayer? The large increase in membrane resistance after your protein incorporation suggested a dramatic change in membrane properties. Yeah,
that's a really good question too. We don't actually fully understand. So when I show the mobility here, we're actually limiting the protein expression a little bit so we can see single molecules. If you let this run longer, you'll see the entire surface covered. So we actually don't have a good number coming out of what the actual density is. I imagine it is pretty high, but this is because we can sort of tune the reaction time in the cell-free environment. You can sort of select what you want to use. So in this case, the density is low, so we can see single molecule tracking. Like you said, with the activity readout, we did have a large increase, so we might have more there. One thing that would be interesting to do is couple this with the QCMD data, and that would sort of give us a better readout of maybe we can actually measure the mass of the protein. But yeah, really interesting question.
Okay, well, with that, thank you very much, Dr. Manzer. That was great. And we look forward, thank you to both of our speakers, and we look forward to seeing you next week.